Hello everyone. This is E-Content Development Program by Department of Higher Education Haryana. Today we'll cover the topic a linkage and recombination from class BSc First Medical Second Sem. Subject Zoology. Myself Seema, Assistant Professor of Zoology from DPG Government College, Panipat. Now firstly we should know what is linkage. Linkage may be defined as tendency of the gene to remain together during the process of inheritance. Means if there are two or three genes, they remain together and they enter into the offspring together. This principle of linkage was first discovered by Bateson and Puneet. However, the term linkage was given by T.H. Morgan. Now some theories are there which are associated with this linkage. First theory was given by Sutton in 1903. According to him, there are number of genes, they are present on the chromosome, and if the number of the chromosome they increases, the number of the gene also increases. For example, if we take the example of human being, we are having 46 chromosome and we are having 25,000 to 30,000 genes on it. However, he could not explain it by any genetic experiment. The second theory, coupling and repulsion theory hypothesis, was given by Bateson and Puneet in 1906. They had so many dihybrid cross among sweet pea, that is Lathyrus odoratus. We have taken one of their work in which they had a cross between blue flower and rounded pollen and red flower and long pollen. In this flower, you can observe that blue flower is the dominating gene, and rounded pollen, that is recessive gene. Means one dominating gene is there, one recessive gene is there in this parent, and in the second parent also. But you will observe that red flower and long pollen is there. Red flower is recessive gene and long pollen is dominating gene. So they have selected two parents with one dominating and one recessive gene are there. In F1 generation, as expected, all the dominating gene, that is blue flower and long pollen, are observed in F1 generation. When they had a cross between this heterozygous F1 generation plants with the recessive parent, that is having red flower and the rounded pollen. Here you can see small b, small l, small l. So they had a cross between the hybrid they grow goat and the recessive parent. What they get four type of individual, and their expected ratio was 1:1:1:1, because according to Mendel, what we have read that we got 1:1:1:1 when we breed recessive parent with the heterozygous offspring. What we observed here, we got 1:7:7:1 instead of getting 1:1:1:1. Here we can observe that 7 ratio is for blue and the rounded pollen, that is parental combination, blue flower and the rounded pollen. Here you can see blue and the rounded, that means 7 ratio is for the parental combination. And here also a red flower and the long pollen. Here you can see in the other parent, red flower and the long pollen were there, and 1:1 for the new combination, that is blue and the long pollen, round and the red and the round pollen. Now what does that mean? That means genetic coupling. Genetic coupling means the gene from the same parent, they try to remain together, that is genetic coupling. And the gene from the different parent, they try to remain away from each other, that is genetic repulsion. In easy word, genetic coupling means the genes from the same parent, they try to remain together. And repulsion means the genes from the different parent, they try to remain away from each other. Now Morgan hypothesis of linkage. This photograph is of T.H. Morgan. He had so many work on Drosophila, and what he suggested that the linked gene, they remain together and they are located on the same chromosome. that is a big thing, that he said the genes are located on the same chromosome, and he got Nobel Prize for his work in 1933. He suggested he suggested the theory of linear arrangement of gene in the chromosome, that on the chromosome genes are linearly arranged on that. Here we have taken one example of Morgan work on Drosophila. Here you can see he had selected male and female. In female you can see here white and miniature wing. These two characters are recessive one, and he has selected one male having wild type, means red eye, and the normal wing it is having. And in the F1 generation what he got a female having red eye and the normal wing. And here you can see he got in F1 generation he got a male having both recessive character. When he allowed them to interbreed among themselves, he got so many type of combination. Here you can see a total progeny he got, as you can see here, was 2441, and out of them he got more percentage of parental phenotype. Parental phenotype means those were having both recessive character or both having the dominating one, that is parental combination. And the offspring he got was 1541. And on the other hand, new combination was appeared to him, but the phenotype was only 900 organisms were obtained with the new combination, means one dominating and one recessive phenotype was there, as you can observe here also. White eye and the wild type wings, one dominating character and one recessive. White eye is a recessive character and wild type wing is dominating character. On the other hand, organism having wild type eye, red eyes with the miniature wing, wild eyes was dominating gene and miniature wing is recessive wing. So by this experiment he concluded that genes, parental type genes, means genes from the same parent, they try to remain together. And new type of combination means genes from the other parent, their combination, their organism having new type of recombination, their percentage is very low as compared to the parental one. Here we can observe in this diagram. Now T.H. Morgan proposed two type of gene arrangement, that is cis arrangement and the trans arrangement. In cis arrangement, as you can see, the both gene, normal gene, they lie on the same homologous chromosome. Here you can see in cis arrangement both dominating A and B genes are located on the same homology chromosome, and on the other hand, on the other homologous chromosome, small a and small b, that means recessive genes are there. In the trans arrangement, as you can observe here, one dominating and one recessive gene are there on the homologous chromosome. So he proposed two type of gene arrangement. Now, the linkage group hypothesis given by Sutton. He said all the genes in a particular chromosome form a linkage group. That means we are having 46 chromosome, means 23 pairs. 23 pairs. So, we are having 23 linkage group. We are taking one more example. For example, maize have 10 pair of chromosomes. So, there are 10 linkage group. Now kind of linkages, there are two kind of linkages. One is complete linkage and second is incomplete linkage. In complete linkage, two or more character are inherited together for more than two generations. Means if there are two genes, they remain together for more than two generations. Means there is no crossing over between them. However, this phenomenon is very rare because some sort of crossing over always occur between them. One we are having the case of male of Drosophila in this. Now in incomplete linkage, there is accidental or operational breakage of chromosomal segment, and this is accompanied by rejoining. That means crossing over occur. This is the natural phenomena. Some sort of crossing over always occur between the chromosomes. Now what is crossing over? Crossing over may be defined as interchange of the corresponding chromosomal part between non-sister chromatid of a homologous pair of chromosome, resulting in the recombination of the gene. What does that mean? That means if there are same chromosome, same homologous chromosome, there is always a exchange of the segment of the genes in between them. Now we will see this what is crossing over, what are the stages of crossing over in the next slides. Now crossing over. Where crossing over started? Crossing over occur in the meiosis and it starts in the zygote stage. And Dotke and Lindgren, they worked on Neurospora crassa, that is pink bread mold. And what they have observed, they observed 8 type of ascospores were observed, and in the ratio of 2:2:2:2. It can be possible if the crossing over occur at the tetrad stage. This was a work by them on the Neurospora crassa, and in this you can observe 2 different type of strains are there in the parents. And if there is no crossing over occur, for 8 type of ascospores they are formed, and they all having the parental genes, means there is no crossing over, they will not get any type of recombination. In the second one, if there is crossing over, if there is exchange of the genetic material between the non-sister chromatid, you will get new combination, non-parental type, you will get new combination, you will get 8 ascospore in ratio 2:2:2:2. This could be happen if the crossing over occur at this stage. Here you can see the tetrad stage is there, means 4 strand you can observe, 2 from the each chromosome. So crossing over occur at tetrad stage, not at the dyad stage. If it would have had happened at the dyad stage, they will get 8 ascospore in the ratio of 4:4. But they got the ascospore in ratio of 2:2:2:2. That means the crossing over would that crossing over occur at the tetrad stage. Now, type of crossing over. There are 2 type of crossing over. First is germinal, that is meiotic crossing over. In meiotic crossing over, the crossing over occur in the reproductive organ of an organism. This is having a great genetic significance because this type of recombination they enter into the offspring. The second one is somatic, that is mitotic crossing over. This occur in the other part of the body other than reproductive organ, but it is not having any great significance because these type of new recombination would not have been able to enter into the other next generation. Now example of crossing over, we are taking one more example in Drosophila fruit fly. Here we have taken 2 individual. One is having dominating type, wild type, and other is having vestigial, black body and vestigial. As you can see here, both recessive character are there, black body and the vestigial wings. And in the other one you can see here both dominating character are there, that is red eye and the normal wing. When they had cross between them, they got what they got, got 4 type of offspring. And in this 4 type, here you can see 2 parental phenotypes were observed and 2 recombinant phenotypes were observed. In the parental phenotype, the number of the individual they got was very high. As you can see, 944 they got with the black and the vestigial, means both recessive character as in the parent type. And they got 965 organism having the wild type, both dominating character as that was in the parent. So they got more parental phenotype. On the other hand, here we can see the recombinant phenotypes, they were very less. The gray and the vestigial means new type of recombination where one is dominating character and one is recessive character, and the other organism is having black body and the normal wing. One is recessive character, that is black body, and one is normal wings, that is dominating one. So recombination phenotype is always less than the parental type. Now the mechanism of the crossing over. How this crossing over occur? How the genetic material are exchanged between the chromosomes, non-sister chromosomes. Now this mechanism involve 4 major steps. The first one is synapsis, then duplication of the chromosome, then crossing over, last is terminalization. In synapsis, here you can see the during synapsis zygote stage, both maternal and parental. Here you can see one chromosome is maternal and one chromosome is paternal. They come together and they are brought together by the mutual attraction between their allelic gene, and during synapsis a synaptonymal complex is also formed that make them to held together during the crossing over. And here you can see in meiosis 2, the second step is duplication of the chromosome. In duplication of the chromosome, dyad become tetrad. Earlier the chromosome is having 2 strands, and in tetrad stage, here you can observe 4 threads are there, that is tetrad stage. So the second stage is duplication of the chromosome where tetrads are formed. Third one is crossing over. In the packetin stage, crossing over the stage is packetin. In packetin there occur chiasmata formation. The point, this is the point where the two the non-sister chromatid they meet to each other, that is a chiasmata. And at packetin stage this chiasmata formation occur, and after chiasmata formation the part of the chromosome they cleaved by a enzyme called as endonuclease. So endonuclease it cleave the chiasmata, the part where they are meeting, and after that they exchange the genetic material. Here you can see the genetic material between blue and the red chromosome, they are exchanged, and this is accompanied by the action of the another enzyme, that is ligase. Ligase means it join the chromosome, and now crossing over is completed over here, and followed by our fourth step, that is terminalization, after which the two non-sister chromatid they repel each other, because so the crossing over involves these 4 steps: synapsis, duplication of the chromosome, crossing over, and terminalization. Now cytological basis of crossing over. For cytological basis of crossing over, one experiment was conducted by C. Stern in 1931. He have taken a special type of female. Here you can see this female is having 2 type of X chromosome. One is having dominating character and one is having recessive character. Dominating character means that female is having red eye and the rounded eye, and on the other chromosome that is carnation. Carnation means less red eye with bar eye. One X chromosome is having dominating character and one is having recessive character, but the special thing is that to that X chromosome a small part of Y chromosome was also attached to that, so that he could easily observe that under the microscope he has chosen this female. Now on the other hand, he has taken the male with X and Y chromosome with carnation eye and the rounded eye. Here you can see one dominating and one recessive gene is there. Now here, forget that part. Now here he got so many type of females, 4 type of female he got in the offspring, and the special thing is that here you can see parental combination of carnation and the bar eye, red eye and the rounded eye, he got them in the parental combination, and in the recombinant one he got carnation and the bar eye, and on the other hand red and the bar eye. And in this experiment also he got more percentage of parental combination as compared to the recombinant one. Okay, in the next slide, kind of crossing over. There are 3 kind of crossing over on the basis of the chiasmata formation. Single crossing over. In single crossing over only one chiasmata is formed. Chiasmata is a point where the 2 homologous chromosomes they meet. In the single crossing over only one chiasmata is there, and here we have the diagram of double crossing over where 2 chiasmata, as you can see in the figure, 2 chiasmata are there. This is the example of double crossing over. And in the multiple crossing over more than 2 chiasmata are formed. More than 2, if there are more than 2 chiasmata are formed, this comes under multiple crossing over. So there are 3 type of crossing over: single, double and multiple crossing over. Now factor affecting crossing over. What are the factors that affect the crossing over? First is the sex. As we observed that generally female are having more crossing over percentage as compared to the male, but as exception are always there, there is high rate of crossing over in male pigeon. In inversion, if there is part of the chromosome is inverted, the less will be the probability of crossing over. Next is mutation. Due to mutation, there is less probability of crossing over occur there. Now age. We see that with the advancement of age, the crossing over it also increases. Next point is radiation. If the cell is exposed to the, if the body is exposed to the X-ray, the frequency of crossing over it also increases. Next is the temperature. In Drosophila, what we had seen that the high and the low temperature, it increases the crossing over. Now the next point is position. In the chromosome, the area near the centromere and the telomere, there is less percentage of crossing over. Now position means the area near the centromere and the telomere. Telomere means end of the chromosome. There is less chances of crossing over. And last is chiasmata. If there is chiasmata, as you can see in the figure, chiasmata where the two chromosomes they meet each other. If there is chiasmata formation, the segment near it, there will be less probability of one more chiasmata formation due to the physical factor. As you can see, if there one chiasmata is there, the probability of the formation of the next chiasmata near it will be, near its vicinity will be very difficult due to physical factor. Now genetic map of crossing over. We can construct genetic map by having knowledge about the crossing over, frequency of the crossing over. This is used to make your genetic or chromosomal maps. Now significance of crossing over, as we know that due to crossing over, this is the proof of the linear arrangement of genes, means genes are linearly arranged on the chromosome and they help in the formation of the genetic map. This crossing over help in the process of evolution, and the plant breeder, they got help by the crossing over. They were able to get a new variety of plant having gregarious growth of fruits. Now genetic map. What is genetic mapping or chromosomal mapping? It is defined as a diagrammatical representation of the genes by their relative distance between them, and we find out this distance between the gene by the their recombination, this crossing over percentage. Now mechanism of crossing over. Here we are chromosomal mapping. Here we are taking one example. Suppose there are three genes on a chromosome, X, Y and Z. Three genes are there on a chromosome. Now we have to construct one chromosomal map. For that we know that for X and Y there is a crossing over percentage is 5. So here on the diagram we have marked X and Y 5 unit apart. After that, what would be the position of the Z? We need to know the crossing over percentage between X and Z and Z and Y. So for the location, for finding out the location of the Z, we should know the what is the crossing over percentage of Z and Y and X and Z. By that we can construct this map. Suppose in this example we have taken the crossing over percentage of X and Z is 3.5. so we make the position of Z between X and Y. Here you can see in the diagram also, X and Z are 3.5 unit apart and Z and Y are 1.5 unit apart. So in this manner we construct a chromosomal or genetic map. Now two terms are there, interference and coincidence. What is interference? As we discussed earlier that when there is chiasmata formation on a chromosome, up to a certain limit no more chiasmata are formed due to the physical factor. As we have seen in the diagram also, here in the chromosome diagram, here when there is already one chiasmata is formed, there will be less probability to up to certain extent no other chiasmata would be able to form because of the physical factor. Now this is called interference. So interference, now interference, it decreases the crossing over because of the chiasmata formation. Now coincidence is the second term. Coincidence is actually opposite to the interference. What is coincidence means? Coincidence means actual number of double crossing over upon expected number of the double crossing over. What is coincidence? Actual number of the double crossing over upon expected number of the double crossing over. Here these, as we know that interference and coincidence, they are opposite to each other. If interference it increases, the coincidence decreases, and if coincidence decreases increases, the interference decreases. Now the value of the coincidence, it vary between 0 to 1. Now importance of the chromosomal map. What is the importance of the chromosomal map? It displays the exact location of the gene. Here we have taken the example of the how we construct a chromosomal map. Now because of the chromosomal map, we got the exact location of the gene and the linkage group in the on the chromosome, and we get specific genes are present. We got this idea that specific genes are present on a specific chromosome, and gene are located at specific location on the chromosome, and this gave the linear arrangement of the gene. Here we are we have thoroughly revised the linkage and the recombination chapter. We have some MCQs regarding this chapter. First one is, who coined the term linkage? Coran, Mendel, Morgan, or De Vries? Now the answer is, who coined the term linkage was Morgan. Now the second question is, how many linkage group of chromosome will be present in case of maize? Is that 5, 10, 15 or 100? The answer is 10, because 10 pair of chromosome are there in the maize. Now the third question is, crossing over in the diploid organism is responsible for dominance of the gene, segregation of the allele, recombination of the linkage between the gene. Now the crossing over occur in the diploid organism is responsible for, the answer is recombination of the linked gene. Now the fourth question is, linkage prevent homozygous condition, segregation of the allele, hybrid formation, heterozygous condition. Now the answer is linkage prevent segregation of the allele. Now the fifth question is, crossing over is more frequent in male, female, both, none of this. Generally crossing over is more frequent in female. Now the next question is, crossing over occur during the stage of packaging, diplotin, diakinesis, zygotin. The answer is crossing over occur during packaging. Now the next question is, number of linkage group in Drosophila is 4, 8, 6 or 10. The answer is, the number of the linkage group in Drosophila is 4, as Drosophila is having 4 pair of chromosome. Now the next question is, ascospore arrangement in Neurospora is 2 by 2, 4 by 4, 3 by 3, 5 by 5. So the answer is, ascospore arrangement in Neurospora is 2 by 2, as we got ratio of ascospores 2:2:2:2 in the offspring. So these are the references from which we have taken the material. And with this, thank you everyone for your patience listening. Thank you.